Hi and Assalamualaikum. My name is Nuraida binti Zamri from AS2576B. This is my FSG660 Project 2 titled The Study on Biofilm Formation of Staphylococcus aureus Isolated from Human Skin under Supervisor Dr. Azia Abdul Aziz. Staphylococcus aureus is a gram-positive staphylococci. They are known to be commensals and harmless towards the human body. However, upon the opportunity to overcome the skin barrier, S. aureus can enter the body and cause infections. One of the major virulent factors of S. aureus in causing infection is the ability to form biofilm. S. aureus is a commensals bacteria on the skin. It is permanently colonized the nasals. However, they can invade the skin through the break of the skin barrier. It can cause infections due to ability to form biofilm and resist towards antibiotic. These studies focus on the ability of isolated S. aureus to produce biofilms and resist towards the common antibiotics. Can S. aureus from the commensals have the ability to form biofilm and be virulent? Are the commensals S. aureus able to resist killing by antibiotic tested? The objectives of these studies are to isolate S. aureus from the commensals of the healthy adults, to determine the ability of isolated S. aureus to form biofilms phenotypically, to determine the antibiotic profiles of the isolated S. aureus, and to confirm the identity of the selected S. aureus by certain rRNA gene sequencing method. Significance of the study: S. aureus are known as commensals on the human skin, so they are safe, but there are the possibilities that these safe bacteria also can cause biofilm formation, which is the virulent factors. Besides that, the S. aureus may also have the ability to be killed by common antibiotic used against S. aureus infections. The studies can contribute to help a better understanding on the virulence factors of commensals S. aureus and better ways to manage this bacteria. The external barrier between the human body and the environment is the skin. Skin dominated by the members of the Staphylococcus, Corynebacterium and Streptococcus. Focus on the nasals, as more than 60% of the S. aureus in the nose are found vestibulumnesi. The first S. aureus infection is impetigo. Impetigo started as a red itchy sore as it heals and crusty yellow or honey color. Second, pneumonia, a form of acute respiratory infections that affects the lungs. 3. Cellulitis, skin infections that can cause by redness, swelling and pain in the infected area. 4th, sinusite, an inflammation or swelling of the tissue lining the sinuses. Virulence factors of S. aureus. Number 1, protein A, capture IgG molecules and prevent phagocytosis. Number 2, coagulase. The clumping factors act directly on the fibrinogen in the plasma, resulting in clumping. The third one is the biofilm formation, which is a community of organisms to the adherence to the surface and form a protective extracellular matrix. S. aureus proteins, including fibrinogen binding protein and surface binding protein A, enable S. aureus to form biofilms. Attachment, multiplication, maturation, and dispersal are the 4 stages of the biofilm formation. For the antibiotic profiles, penicillin first introduced to S. aureus and become resistant after a period of time. Methicillins then introduced to this bacteria and also become resistant towards the antibiotics. Method 1, S. aureus isolation. Methods 2, 96 well microtiter plates biofilm assay. Method 3, antibiotic profiles. Method 4, amplification 16S rRNA. 10 presumptive S. aureus that shows a yellow colony on MSA were successfully isolated. There were gram-positive cocci in cluster, catalase positive and coagulase positive. This result indicates the general characteristics of S. aureus. Biofilm assay was conducted on microtiter plate with S. epidermidis ATCC 35984 and S. epidermidis ATCC 122228 were used as positive and negative control. From the 10 samples, 6 show moderate biofilm formation. There were SA2, SA3, SA4, SA5, SA7 and SA8. The remaining are the weak biofilm former. All 10 presumptive SRS isolated were resistant against cefoxitin, penicillin, ampicillin, and vancomycin. However, for ciprofloxacin, only SA8 were resistant to this antibiotic, while the remaining 9 isolates were susceptible. For erythromycin, 2 isolates SA7 and SA8 were resistant, while other 8 were susceptible to these antibiotics. In conclusion, SA7 and SA8 are multi-drug resistant since they are resistant to all the antibiotics, so these 2 samples was chosen for further identification. From the results of amplification, SA7 and SA8 was confirmed as SRS. In this study, the SRS collected from the healthy individuals indicate that SRS colonized in the nasals of every individual. It is dominantly colonized the nasals. SRS should be considered as a harmless commensals. However, upon breaking the skin barrier, SRS can enter the body and cause infections. SRS causes infection as it has the ability to form biofilm, which are one of the major virulent factors. SRS from the nasal can cause biofilm as the majority of the SRS isolated have the ability to form biofilm. The antibiotic susceptibility test results reveal that the commensals SRS can be resistant to multiple drugs. SRS isolates were also resistant against the antibiotic tested in the study. 2 isolates, SA7 and SA8, were MDR because they were resistant to 3 classes of antibiotics. This shows that besides being commensals, not only they can form biofilm, but they are also MDR. Its multi-drug resistant phenotypes make it difficult for treatment. In this study, 10 presumptive SRS has been successfully isolated. From the 10 isolates, 6 were found to be moderate biofilm former, while the remaining 4 produced weak biofilm. This shows that although these SRS were commensals, they are possess one of the main virulence factors of SRS, which is the ability to form biofilm. All the 10 isolates SRS were found to be resistant against cefoxitin, penicillin, ampicillin, and vancomycin, while majority of the isolates were sensitive against ciprofloxacin and erythromycin. SA7 and SA8 were found to be MDR. Interestingly, these 2 isolates also the moderates of biofilm former. This suggests that isolates were commensals and supposed to be harmless, but they also have the virulent factors, which indicates that they are able to penetrate the skins and they can cause serious infections and also resist to the antibiotics, make it difficult to treat. The result contribute valuable insights to the broader understanding of SRS virulent factors and their implication for clinical treatments. For recommendation, this study should be conducted with larger samples of SRS isolates for better understanding, and also challenge the SRS isolates with many types of antibiotics. That's all for me. Thank you for listening.